Started. Good evening, and thank you all for attending the CALA seminar series. Before we get started to introduce our speaker, we're just going to make an announcement that we're going to have a 2023 ATS CALA gathering, presumably on the Saturday, May 20 night. So if you plan to attend ATS meeting this year, please fill out the form Ke sent out. Also, we are going to award have a couple of awards this year. So we're going to award one establishing investigator, one junior investigator, and one trainee award from CALA meeting this year. So please nominate yourself, your colleagues, or your trainees for the award, and send your nomination to Dr. Jianwen Que in Columbia University through this email, and by Friday, March the 17th. So I will put this information in the chat, so you may can get the email address and also the link again from the chat. So that is our housekeeping stuff. So now I'm going to introduce today's speaker. Today's speaker is Dr. Bibo Zhu from Huazhong Agricultural University. So Bibo did his bachelor, master, and PhD in Huazhong Agricultural University. And then after his PhD, I have the fortune to recruit him to my lab initially when we were still in Indiana University, and then he moved with me to a Mayo Clinic, and last year we moved from Mayo Clinic to UVA. So Bibo then went back to Huazhong Agricultural University because he liked the university so much, and become a professor in the School of Veterinary Medicine. During his 6 years, a little bit more than 6 years in my lab, Bibo is very productive in my lab, and I'm really fortunate to have him in my lab. He publishes 3 pretty impactful papers in my lab, and when he has time, he's going to write his 4th paper. So for today's talk, we're going to talk about tissue-resident macrophage function and fate in respiratory virus infection. So Bibo, the stage is yours.
Thank you so much, Jie, for your kind introduction. Thank you for having me to present our work here. So today, my talk is tissue-resident macrophage function and fate in respiratory viral infection. I would like to share 2 stories which I completed in Dr. Jie Sun Lab during my postdoc training. So my research interests focus on the respiratory viral infection, including influenza virus and SARS-CoV-2 and host immunity. The respiratory viral infections cause viral pneumonia. SARS-CoV-2 in particular poses a great burden to the society, killed almost 7 million people all over the world till now. So it's quite important to understand the pathophysiology and the protective immune response to respiratory viral infection for the development of future preventive and therapeutic means. This image shows the respiratory viral infection and host immunity against the virus infection. At the very early stage, virus replicates reach the peak level at 4 to 6 days post-infection, and then will be cleared by the host antiviral responses within 8 days. At the early stage, the virus replication will activate the innate immune responses, which limits virus replication as well as initiate the cellular or humoral responses. The adaptive immune responses further clear the viral particle as well as the viral infected cells. So after virus being cleared, the innate immune responses will return to the homeostasis. The host will show mild disease. However, when the innate immune responses persist, the host will show severe disease due to the immune responses induced tissue damage. Therefore, the innate immunity is a double sword. On the one hand, the innate immunity act as the front line to defend the microbial infection. They also promote the tissue repair. However, on the other hand, the uncontrolled immunity will induce the inflammation, mediates the tissue damage, which play detrimental roles to the host. However, how to balance the protective function versus the deleterious effect of innate immunity is not well studied, especially in the respiratory viral infection. The underlying cellular and molecular mechanisms is also largely unknown. My research focuses on the innate cells, macrophages. So my first part is to understand the transcriptional regulation of tissue-resident macrophage function and fate following respiratory viral infection. So macrophages can be divided into two groups. Some macrophages come from bone marrow called monocyte-derived macrophages. Those macrophages are well studied in vitro. For instance, those monocytes can be differentiated into classical M1 or M2 macrophages with different stimulars. Some other macrophages come from the yolk sac or fetal liver. Those macrophages can park in the tissue during adult, called tissue-resident macrophages. Those macrophages can self-renew with minimum monocyte contribution during homeostasis. I'm interested in the non-resident macrophages. It's also called alveolar macrophages, which locate in the alveolus. Those macrophages can self-renew and promote non-development. Those alveolar macrophages are the first immune cells contacting microbial, so it can activate the immune responses locally and initiate the adaptive immune responses. Also, those alveolar macrophages can promote the tissue repair as well as the regeneration. So those alveolar macrophages are multifunctional immune cells. How about the heterogeneity? Our groups, as well as others, did a single-cell RNA-seq to look at the alveolar macrophages heterogeneity. One Ph.D. student, Yue, in our lab, found 6 clusters in the alveolar macrophages during homeostasis based on their molecular marker. The cluster 3 are more like inflammatory macrophages because they highly express the pro-inflammatory cytokine genes. Also, the cluster 4 and the cluster 5 express high level of proliferative or self-renewal related genes. We are very interested in these two these two subsets. Currently, most of the paper study either the macrophage function regulation during homeostasis or viral infection, or studying the macrophage development, differentiation, and self-renewal. Fewer papers study how this macrophage function as well as self-renewal, the relationship as well as the significance during respiratory viral infection. So we asked the question, what's the relationship between macrophage inflammation and self-renewal? How did they transcriptional regulated by those transcriptional factors during respiratory viral infection, and what's their significance during virus infection? Firstly, we profiled the transcription factors in available macrophages following influenza virus infection. So virus infection could upregulate CTMB1 transcription factor. This transcription factor encodes beta-catenin. So what's Wnt beta-catenin signaling? This image shows the canonical Wnt beta-catenin signaling pathway. Without Wnt, beta-catenin can be phosphorylated by GSK3 beta and subject to degraded by the proteome of the proteasome. When the Wnt exists, Wnt ligand will bind to the receptor and promote the release of beta-catenin from the complex. Beta-catenin will interact with cofactors TCF1 or others to regulate downstream gene expression. Those Wnt beta-catenin signaling could participate in regulating cell proliferation, the cell inflammation, stimulus, as well as metabolisms, etc. So we are wondering whether Wnt beta-catenin signaling could regulate available macrophage function and self-renewal following virus infection. To answer this question, we first needed a single-cell RNA-seq in available macrophages in vitro following Wnt3A treatment. So our data suggested that Wnt3A treated available macrophages are quite different from the control treated available macrophages. The inflammation-related genes were enriched in Wnt3A treated available macrophages. However, self-renewal related genes, such as the embryonic stem cell module related genes, enriched in the control group. Therefore, we conclude that in vitro, Wnt beta-catenin signaling promotes macrophage inflammation and inhibit the macrophage proliferation or self-renewal. How about in vivo? What's the significance for this phenomena? So in vivo, we generated the myeloid beta-catenin deficient mice and sorted available macrophage from those wild-type or beta-catenin deficient mice at different time points following influenza virus infection. We found beta-catenin deficiency significantly decreased inflammation-related gene expression. Furthermore, beta-catenin deficient mice are more resistant to virus infection. Therefore, we found that Wnt beta-catenin signaling promote macrophage inflammation in vivo. How about this part? it's known that during respiratory viral infection, the available macrophages numbers decreased at an early time point and then repopulates. We also found beta-catenin deficiency increased available macrophage repopulation. So the available macrophage decrease could be due to the inflammation-induced paraptosis or apoptosis. However, how available macrophage repopulates during the late time points is debated area. Some groups suggested that monocyte differentiation may contribute to available macrophage niche. However, other groups suggested that the survival available macrophage can self-renew and fill the available macrophage numbers. To figure out whether monocyte differentiation could contribute to available macrophage repopulation in our system, we did parabiosis by joining CD45.2 and CD45.1 wild-type mice together for 4 weeks, and then infected both parabiotic mice with influenza virus. So after 15 days, we checked the cell types. For circulating cells, for instance, monocyte CD4 T cells, these cells can circulate between the parabiotic mice. About 50% of the cells come from the parabiotic mice. For available macrophages, undetectable cells from the parabiotic mice. So this data suggested that monocytes are not the major contributor to available macrophage repopulation. And I would like to point out that we just checked the day 15 data. Some papers suggested that at the late time point, such as day 30 or even later, monocyte could differentiate into available macrophages during virus infection or bacterial infection. So then we checked whether the available macrophage can proliferate locally. So we found about 11% of the wild-type available macrophages proliferation, and the beta-catenin deficiency enhanced the proliferation rate. This is consistent with previous data that beta-catenin deficiency enhanced available macrophage repopulation. Therefore, our data suggested that available macrophage can proliferate locally and contribute to available macrophage repopulation. So then we conclude in vivo that Wnt beta-catenin signaling promotes available macrophage inflammation and inhibits macrophage proliferation and self-renewal. We ask the next question: what's the mechanism? So our single-cell RNA-seq data suggests that Wnt3a Wnt3a significantly enhanced HIF-alpha expression in available macrophages. And also one paper suggested that beta-catenin and HIF-alpha interaction under the hypoxia condition. We assumed that Wnt beta-catenin signaling regulates macrophage function and self-renewal through interacting with HIF-alpha. To answer this question, we examined the interaction between HIF-alpha and beta-catenin following Wnt3a treatment. So without Wnt3a, beta-catenin interact with the classical cofactor TCF4. Following Wnt3a treatment, beta-catenin interacts with HIF1 alpha. Furthermore, we look at the inflammation or self-renewal related gene expression in wild type or beta-catenin HIF1 alpha deficient alveolar macrophages following Wnt3 treatment. So Wnt3 treatment significantly enhanced inflammatory related gene expression, while inhibited self-renewal gene expression in wild type alveolar macrophages. The beta-catenin deficiency or HIF1 alpha deficiency significantly rescue those phenomena. So this data suggested that Wnt beta-catenin signaling regulates macrophage inflammation and self-renewal through HIF1 alpha. It's known that HIF1 alpha regulates macrophage metabolisms. So we examined the macrophage metabolic status by seahorse in vitro. Wnt3A treatment significantly enhanced macrophage glycolysis, while decreased macrophage oxphores through beta-catenin and HIF1 alpha. As we know, the oxphores always reflect mitochondrial function. So we examine the mitochondrial morphology by the electron microscopy. Wnt3A treatment enhanced the mitochondrial damage in alveolar macrophages. However, in beta-catenin or HIF1 alpha deficient alveolar macrophages, the abnormal mitochondrial levels similar as the wild type without Wnt treatment. This suggested that Wnt beta-catenin's HIF1 alpha axis regulate alveolar macrophage metabolic status. So we asked if the metabolic shift could regulate macrophage inflammation and proliferation. We block the macrophage glycolysis and mitochondrial function by inhibitors. So 2DG inhibition of glycolysis significantly inhibited the macrophage inflammation, but didn't affect macrophage proliferation. Well, when we inhibited the mitochondrial complex 1, 2, or 3 by inhibitors, significantly inhibited macrophage proliferation. Therefore, our metabolic mechanisms are Wnt beta-catenin. Wnt treatment significantly enhance beta-catenin interaction with HIF1 alpha. This interaction promotes macrophage inflammation, depends on glycolysis, and this complex also inhibited the macrophage self-renewal by inhibiting mitochondrial function. So we found the molecular mechanism how Wnt beta-catenin regulate macrophage inflammation and self-renewal. The next question is, what's the cellular mechanisms? Are those inflammatory macrophages and the self-renewal macrophages different in vivo? We got HIF1 alpha report mice, which called EJN3 YLP mice from Dr. Ananda lab. We sorted out YLP high and YLP low alveolar macrophages. So from flow cytometry, YLP high alveolar macrophages express high level of HIF1 alpha and active beta-catenin. And those YLP high alveolar macrophages express high level of cytokine related genes. Well, express low level of tissue repair or self-renewal related genes. Furthermore, YLP low available macrophages show high potential proliferation compared to YLP high available macrophages. Therefore, we conclude that beta-catenin high and beta-catenin low available macrophages are inflammatory available macrophages, where beta-catenin low and HIF alpha low available macrophages are proliferative self-renewal macrophages. So those inflammatory macrophages are good for anti-microbe infection, but bad for the excessive inflammation. Those self-renewal or proliferation of the macrophages are good for the macrophage homeostasis and the host recovery. Our main finding for this study is that the macrophage inflammatory activity and self-renewal are uncoupled. This phenomena is good for preventing the chronic inflammation as well as promoting tissue repair. So this is the first part for this talk. Our previous data suggested that macrophage regulates influenza pathogenesis. More clinical data or the lab data suggested that macrophages also contribute to COVID-19 induced inflammation. Therefore, we asked if we could treat the viral pneumonia, including COVID-19, by manipulating macrophage function. This is my second part. Our first paper suggested that macrophage inflammation depends on glycolysis. So whether we could target glycolysis to regulate macrophage inflammation. This image shows the glycolysis metabolisms. So from glycolysis to lactate, this part is well studied in vitro in inflammatory macrophages. However, mitochondrial pyruvate carrier mediates pyruvyl metabolism in mitochondria in available macrophages following respiratory viral infection is not well studied. To check whether MPC mediated pyruvate metabolism could regulate virus infection, we generated myeloid MPC deficient mice. In this mice, the MPC deficiency diminished pyruvate metabolism in mitochondria. These deficient mice are more resistant to influenza virus infection, suggesting MPC-mediated pyruvate metabolism regulate influenza pathogenesis. So there is a drug called MSDC0602K, which is generated by the company Serious Therapeutics. It belongs to TZD family, and it's a new insulin sensitizer that can attenuate the uptake of pyruvate by the mitochondria. So the company finished phase 2B clinical trial for this drug, showed positive effects on glycemic control in type 2 diabetes. Since this drug is MPC inhibitor, we treated influenza virus infected wild mice with vehicle or this drug, MSDC. So the drug treatment significantly protect significantly protect the mice from influenza virus infection without affecting virus titers. Furthermore, we check the efficacy of this drug in SARS-CoV-2 infected mouse model. So MSDC treatment decrease non-inflammation and damage and enhance the mouse weight recovery. So combined all the data, we found MPC deficiency or MPC inhibitor attenuates influenza and COVID-19 diseases. We asked the underlying mechanisms. The drug MSDC was administered through IP, so it's systemic administration. We asked first, which cell types are affected by this drug? We did a single cell RNA-seq in the non-cells from vehicle or MSDC treated mice. This UMAP shows all the cell types in the non-tissue. So MSDC treatment enhanced available macrophage 81, 82 epithelial cells numbers were diminished 96C high monocytes, neutrophil numbers. We are interested in available macrophages, monocytes, as well as monocytes-derived macrophages in this system, because those cells are critical for COVID-19-induced inflammation. So we further analyzed the gene profile of those cells. MSDC treatment diminished a number of inflammation-related gene expression. However, most of those genes didn't change in monocyte-derived macrophages or monocytes. To further confirm the data, we sorted out available macrophages, monocyte-derived macrophages, and the monocyte from vehicle or MSDC treated mice. Similarly, MSDC treatment diminished pro-inflammatory cytokine expression in available macrophages, but not monocyte-derived macrophages or monocytes. Next, we did RNA-seq in vitro in available macrophages following vehicle or MSDC treatment. The RNA-seq data also suggests the cytokine inflammation or antiviral signaling related gene decreased in MSDC treated available macrophages compared to the control treated available macrophages. All this data suggested that MSDC treatment diminished available macrophage inflammation both in vitro and in vivo. So now we know MPC-mediated pyruvate metabolism in mitochondria regulates macrophage inflammation. Our previous study suggests the HIF-1 alpha regulate macrophage inflammation. Therefore, we measure the HIF-1 alpha phosphorylated P65 STAT1 expression in available macrophages in vitro in the presence of vehicle MSDC. So MSDC treatment only decreased HIF-1 alpha expression without affecting P65 or STAT1 phosphorylation. This suggested that HIF-1 alpha may participate in regulating of macrophage inflammation following MSDC treatment. To answer this question, we used HIF-1 alpha activator to treat MSDC treated available macrophages. those two activators could rescue CCL2 or TNF-alpha expression. This data suggested that regulation or reduction of alveolar macrophage inflammation by MSDC through decreased HIF-1 alpha expression. So from this image, we could see that in mitochondria, pyruvate metabolism participated in the TCA cycle. They oxidized in the TCA cycle. So we measured the metabolites of TCA cycle. Interestingly, we found poly-IC-treated alveolar macrophages significantly enhance succinate levels. However, MSDC treatment decreased these succinate levels. However, other metabolites didn't change between vehicle or MSDC treated groups. Previous paper suggested that succinate can stabilize HIF-1 alpha and regulate HIF-1 alpha expression. Then we asked whether succinate involved in our model. So we treated the alveolar macrophages with succinate activator. Those activator DMS could rescue TNF or CCL2 expression in MSDC-treated alveolar macrophages. Further, the activator could enhance HIF-1 alpha expression in MSDC-treated alveolar macrophages. So this image shows the molecular mechanisms by which MSDC dumping macrophage inflammation following respiratory viral infection. So the drug inhibits MPC function, further inhibits pyruvate metabolism in mitochondria. This phenomena decreased succinate levels, resulting in decreased HIF-1 alpha expression, further affect the pro-inflammatory gene expression in alveolar macrophages. So it's well known for individual with metabolic syndrome such as obesity or type 2 diabetes. They are very dangerous to SARS-CoV-2 infection. Also, the clinical data suggested that they are usually show severe disease and poor clinical outcome. Currently, some clinical data suggested that some COVID-19 patients showed a higher blood glucose level and even developed type 2 diabetes. So it's quite important to treat the metabolic condition during COVID-19 in the patients with metabolic conditions. Our previous data suggested that MSDC could regulate macrophage inflammation, and also the clinical trial suggested that MSDC could lower the blood glucose. So we asked if this MSDC could simultaneously mitigate hyperinflammation and hyperglycemia in obese hosts. To this end, we generate diet-induced obese mice and infected those mice with SARS-CoV-2 virus following vehicle and then treated with vehicle or MSDC. Consistent with the clinical data, the DIO mice, all of the DIO mice succumbed to virus infection, while most of the mice are survived for the lean mice. Later, we measured the blood glucose and the cholesterol levels following the vehicle MSDC treatment. The DIO mice showed enhanced blood glucose and cholesterol compared to the lean mice. However, MSDC treatment significantly lower those glucose or cholesterol levels. This suggested that MSDC treatment could maintain the metabolic health. Next, we found MSDC treatment lower the non-inflammation and the non-damage. And also the treatment enhanced the host survival following SARS-CoV-2 infection. However, there are almost more than 60% of the mice died. We guess that this could be due to uncontrolled virus replication. Therefore, we treated the obese mice with MSDC and or antiviral drug. So the drug alone could enhance the host survival. After MSDC treatment, the survival rate significantly increased. So we suggested that the MSDC combined with the antiviral drug could be used to treat obese mice following COVID. Next, we examine whether MSDC play roles in regulating COVID-19 associated inflammation in human samples. We isolated the available macrophages from 3 donors and then infected with SARS-CoV-2 virus in vitro in the presence of vehicle MSDC. Macrophages from 2 donors show significantly enhanced cytokine or chemokine expression compared to the control group. However, MSDC treatment significantly decreased those genes. Furthermore, we treated non-cells from autopsy of COVID-19 patients and checked the pro-inflammatory gene expression. MSDC treatment also decreased some of the pro-inflammatory gene expression in the non-cells. So MSDC diminished COVID-19 associated inflammation in human samples. So our study suggested that the drug MSDC could simultaneously promote host metabolic health and suppress the hyperinflammation caused by respiratory virus by targeting the mitochondrial pyruvate translocation. So we also proposed the molecular mechanisms. We suggested that in clinical treatment of COVID-19 with MSDC or with MSDC plus the antiviral drug would be useful for patients with the metabolic syndrome. So this is the second part. Last but not least, I really appreciate my postdoc mentor, Dr. Jie Sun. He helped me a lot at all aspects. Xiaoqin is the co-first author in the second part. Yue analyzed the single-cell RNA-seq data for me in the first part. I would like to also thank all the members in Jie Sun Lab. Thank all the collaborators and funding. Thank you for your attending. I would like to take questions.